predators our ancestors feared left marks on the landscape. You could see a cave bear's tracks, hear a pack of dire wolves moving through the dark, read the stillness that preceded a large cat's approach. The threats that came from outside the body announced themselves in ways that at least allowed for the possibility of detection, avoidance, and occasionally survival. The threats that came from inside the body were different in every way that mattered. Parasites are among the oldest life forms on Earth. They predate complex animal life by hundreds of millions of years, and they have been refining their techniques for exploiting host biology for longer than most of the animals that would eventually carry them have existed. By the time the first hominids were walking the African savanna, the parasitic organisms that would spend the next several million years living inside human bodies had already been perfecting their craft inside the bodies of our evolutionary predecessors. They didn't need to adapt to humans. We walked into a system that was already built, already optimized, and already waiting. The first thing to understand about ancient parasitic infection is the scale. Modern humans in developed countries with access to clean water, sanitation infrastructure, and pharmaceutical intervention still carry parasitic infections at rates that most people find surprising when they encounter the data. Remove all of those interventions, the clean water, the sanitation, the medicine, and place a human population in close proximity to the water sources, soil conditions, and animal populations of the ancient African environment, and the infection rates become effectively total. Not most people. Not the majority. Everyone, across entire lifetimes, carrying multiple simultaneous infections that modern medicine would treat as emergencies. Schistosomiasis offers one of the clearest windows into what this looked like in practice. The parasitic flatworm responsible for schistosomiasis, Schistosoma, completes part of its life cycle inside freshwater snails and releases larvae into still or slow-moving fresh water. Those larvae are capable of penetrating intact human skin on contact. Not through a wound, not through ingestion, but through the surface of unbroken skin that has been in contact with infected water for as little as a few seconds. For a hominid population that needed to interact with African waterways daily for drinking, fishing, and crossing, avoidance was not a realistic option under any conceivable circumstance. Once inside the body, schistosoma larvae migrate through the bloodstream to the liver, where they mature into adult worms that can live for decades. The adult worms release thousands of eggs daily, most of which become lodged in surrounding tissue, liver, intestinal walls, bladder lining, where the immune response to the eggs causes progressive, irreversible scarring. Chronic schistosomiasis produces an expanding constellation of effects. Severe anemia through sustained blood loss, progressive liver and spleen enlargement to the point of visible external distension, chronic fatigue so profound that sustained physical exertion becomes increasingly difficult, and in advanced cases, complete failure of the affected organ systems. The process is slow enough that infected individuals remain functional for years while the internal damage accumulates, which is part of what made the parasites so effective across evolutionary history. It didn't kill quickly. It diminished gradually, draining the physical capacity of the host over time frame measured in decades rather than days. Paleoparasitological evidence for schistosomiasis in ancient human populations is direct and unambiguous. Calcified schistosoma eggs have been recovered from Egyptian mummies dating back several thousand years, and the distribution of the parasite across ancient populations tracks almost perfectly with proximity to fresh water sources, which is to say, it tracks almost perfectly with wherever ancient human populations chose to live, because those locations were almost always determined by access to fresh water. Guinea worm disease presents a different category of biological horror entirely. The guinea worm, Dracunculus medinensis, enters the human body through the ingestion of contaminated water, containing tiny water fleas that carry the worm's larvae. The larvae are released in the digestive system, penetrate the intestinal wall, and migrate through the body's connective tissue for roughly a year, growing to adult length, which in females can reach up to a meter, before the mature female worm begins migrating toward the skin surface to release larvae into whatever water source the host happens to be standing in. The emergence process is not quick. The worm creates a blister at the skin surface, typically on the lower leg or foot, that ruptures to allow partial emergence. The standard method of removal, which has been documented across cultures for thousands of years and is likely the origin of the rod of Asclepius symbol used in medicine today, involves winding the worm around a small stick, a few millimeters per day over a period of weeks, because attempting to pull the worm out faster risks breaking it inside the body, which causes severe infection and can be fatal. During the emergence period, the affected limb is typically non-functional. For hunter-gatherer communities dependent on physical mobility for survival, a member of the group with a guinea worm emerging from their leg was a member of the group who could not walk, hunt, gather, or defend themselves effectively for the duration of the emergence process. Toxoplasma gondii occupies a different register of horror altogether because its primary effect is not physical deterioration, but neurological. Toxoplasma is a single-cell parasite whose definitive host is the cat family, but which can infect virtually any warm-blooded animal as an intermediate host, including humans. Infection typically occurs through contact with infected soil, contaminated water, or undercooked meat from an infected animal. The parasite forms cysts in the brain and muscle tissue that can persist for the lifetime of the host. In most modern humans with functional immune systems, Toxoplasma infection is asymptomatic or produces only mild initial illness. But research over the past several decades has accumulated significant evidence that chronic Toxoplasma infection produces subtle but measurable behavioral and neurological changes in human hosts, altered risk assessment, reduced reaction time, changes in personality that trend toward reduced caution in potentially dangerous situations. The mechanisms behind these behavioral changes are not fully understood, but they appear to relate to the parasite's influence on dopamine regulation in the brain. In rodent hosts, these behavioral changes are not subtle. Chronically infected rodents lose their instinctive fear of cat odor, a change that is transparently beneficial to the parasite, which needs its host to be eaten by a cat in order to complete its life cycle. For early human populations living in close proximity to large wild felids, lions, leopards, cheetahs, and others, the possibility that widespread Toxoplasma infection was subtly altering the risk assessment behavior of infected individuals introduces an unsettling dimension to the already formidable challenge of prehistoric survival. An ancestor whose amygdala was responding to Toxoplasma-mediated dopamine disruption was an ancestor whose capacity to read and respond to danger signals may have been measurably compromised at precisely the moment when accurate threat assessment was the difference between living and not. The internal parasitic environment of early humans also included intestinal worm infections of a scale that is genuinely difficult for modern people to conceptualize. Ascaris lumbricoides, a large roundworm still infecting an estimated 800 million people globally today, was effectively ubiquitous in ancient human populations. Adult Ascaris worms can reach lengths of 35 centimeters and live in the small intestine in numbers ranging from a few individuals to several hundred. A heavy infection competes directly with the host for nutritional resources, causes chronic malnutrition regardless of food intake, can migrate into the bile ducts and lungs during larval stages causing additional organ damage, and in sufficient numbers can cause intestinal obstruction. For populations already operating on nutritional margins determined by the availability and difficulty of acquiring food, the additional metabolic burden of carrying hundreds of intestinal worms was not a background inconvenience. It was a direct sustained drain on the physical capacity required to survive. Whipworm, hookworm, and threadworm infections operated on similar principles. Chronic, persistent, largely invisible to the infected individual in the early stages, and collectively producing a baseline of reduced physical capacity, chronic anemia, and nutritional compromise that would have been the universal background condition of early human life rather than an exceptional circumstance. What emerges from looking at ancient parasitic infection honestly is a picture of the human body as a contested environment, a landscape that multiple other organisms had learned to exploit, inhabit, and manipulate long before the concept of medicine existed to push back against them. The external predators our ancestors feared were dramatic, visible, and occasionally survivable through skill, coordination, and luck. The internal predators were none of those things. They were invisible until the damage was already done, unkillable by any means available at the time, and in many cases so well adapted to their human hosts that they could persist for the entire lifetime of the infected individual without ever producing symptoms dramatic enough to identify the source of the slow compounding deterioration they caused. The human body that our ancestors were trying to keep alive was not a pristine biological system temporarily threatened by external dangers. It was a system already under sustained biological occupation from within, by organisms that had been refining their techniques for millions of years, that had no evolutionary incentive to stop, and that in many cases are still with us today. Have a happy day.